What you saw was an optical tweezers that can be built for less than 400 euros. In this video, I'm going to show you how you can build your own device. A scientist named Arthur Ashkin was a pioneer in this field. In 1970, he showed that light carries momentum and can apply force to a small object. In 1986, he demonstrated the version of optical tweezers that we use today. After many years of application, especially in biology, he received Nobel Prize in 2018. He's a very important figure in this field, and if you like science stories, I highly recommend to watching his Nobel talk in Aspen. I've put the link in the description below. It might look like a science fiction, a bit like tractor beams in Star Trek. It's amazing that you can move objects just with light. In Ashkin's own description, it's just the focus of a light. With optical tweezers, you can trap anything, biological sample, even single atom. The great feature is that it's very gentle. You can trap single cell, blood cell, bacteria, or sperm cells without damaging them. But the thing is, these objects are usually too small 10 times smaller than human hair, so it's invisible to naked eye. Therefore, you need a microscope to observe them. To build our optical tweezers, we use OpenFlexure platform, which is an open source 3D printed microscope. It's developed by a great community and it's used over the world. It's super stable. There is a lot of documentation that shows you step by step how to build one with around 300 euro. If you go to the microscope tab, you see the 3D view of this design. The link is in the description below, but I want to show you that we use the backport of the microscope to add our laser for the optical tweezers. Here you can see a schematic of the design. The blue part shows the normal microscope light. The red path shows the light for the optical tweezers. In this video, I will show you the design, how to assemble it, the extra part that you need. And we basically need to expand the laser beam with two lenses to fill the rear aperture of the objective lens and integrate it to the main light path. All the 3D printed parts of the extension are here. A laser holder to keep the laser and the first lens, a cube to hold the dichroic mirror, a coupler that connects the laser holder to the cube, a modified OpenFlexure optical module, and an alignment tool. We recommend you to print all the extra parts with support, as we got better results with them. Before we dive into the assembly, I need to talk about something important: laser safety. Never directly look at the laser beam, and please do not start more than 1 milliwatt of laser power. First, clean the printed parts so there is no support or debris. Here we use an 60x objective lens from a Chinese manufacturer that costs around 50 to 60 euro. For the printed parts, it's a good idea to check all the parts fit together properly before going further. The cube, for instance, has a sliding mechanism into the coupler. The other side of the coupler, it's a thread that fits into the laser holder. The laser holder is actually designed by two parallel tubes. The inner tubes that we mount the laser, and the outer tube acts as a reference tube to adjust the laser angle. These two tubes are connected by few flexible joints such that it gives us few degree of control on the alignment. To get a better control over the laser alignment, we use heatsink insert nuts. I actually find it easier to heat the insert nut with a soldering iron and gently push it into the laser holder. You need 3 of them every 120 degrees, and you should not actually push it too much to damage the plastic parts. And the last one. There you go. Now you can check all of them with a screw that they are not blocked or clogged. These are the same types of screw that have been used in the microscope leg and the condenser part. And the same type of thumb screw has been used as well. Now is the time for the optics. We need two lenses and a dichroic mirror. First, we use a 1/2 inch doublet lens with 17mm focal length. They have a curved side and flatter side. And the rule is the curved side should face the collimated beam, basically the incoming laser light. After placing the lens, put the lens cap in and fix it into place. The screws just need to be loose, not pushing anything. Good. Next is the laser. We use a 6mm diameter red laser pointer with a wavelength of 635 nanometer and a maximum of 1 mW. They are pretty cheap, about 50 cents per each. Fix the laser in the holder with the M2 tab screw. The laser should not move anymore and just need to be fixed in the position as well. If the laser doesn't move, the laser holder is ready and we move to the cube. The cube has two slots for the optics, dichroic mirror and a filter. The dichroic mirror one is chosen to match the laser wavelengths. In this case, it reflects red light and transmits blue and green. I found them pretty cheap in AliExpress, about €1 per each. It's good to mention that dichroic mirrors have two sides. The coated side that has a better efficiency, where you can usually find it with a phone light or flashlight. Try to place the dichroic mirror in the middle of light path or in the middle of the holes. We noticed actually quite a lot of back reflection into the camera, so we decided to use two dichroic mirrors. One acts the main mirror and the second one works as a filter. If you can, it's better to use a proper filter actually. Now it's time for the 1/2 inch 50mm focal lens, exactly as described in OpenFlexure high-resolution microscope design. It helps us to collimate our laser light, and as the rule said, the curved side must be the collimated light. So in this case, the curved side needs to be on the top or opposite of the laser direction. We actually do not have so many electronics. Some of these cheap laser modules do not run on exactly 5 volt. With a DC-DC converter, you can adjust the voltage to what laser needs. Optionally, you can place a key to have control on turning on and off the laser. The fun part starts. At first, the laser spot looks off the side. Our goal is to center the laser in the optical module so that the beam enters the back of the objective lens and travels straight along its axis. To do so, we align the beam at 2 points to make sure that the light travels in parallel to the objective lens. To properly control a beam, we usually need 2 degrees of freedom. We found that just moving the laser itself is not enough, so we designed the cube to be slightly flexible too, and with 2 M2 screws, we can adjust the angle of the dichroic mirror. Now we need the alignment tube. Place them in place of the objective lens. With the long tube, you align the beam at the further point, or the cube part with the 2 M2 screws, and then at closer to the lens, you should center the beam with the 3 screws on the laser holder. You at least need to repeat the process a couple of times until you make sure the laser is centered in both short and long tube. For a better precision, you can print alignment tube with a 1mm pinhole and a small cap where you can reach a better accuracy. If the laser passes through both pinholes, your beam is well aligned. Once the alignment is done, you can put the camera in exactly how it is explained in OpenFlexure website. I'm using a 60x objective lens because it's more convenient, but a 100x oil immersion lens gives you a tighter focus of the beam, so a stronger trap. A small fact, objective lenses are color coded, 100x has a white ring and 60x has a blue ring. The objective lens is really the heart of this microscope. The high quality ones from major brand can be very expensive, but we got quite good result with these Chinese infinity corrected plan objectives. For an optical trap, you usually want to have at least numerical aperture 0.8. That gives you a focus spot around 1 micron in size for a red laser. I'm assuming you already have your OpenFlexure microscope built. To make space for the laser holder, you just need to cut away the extra plastic around the side of the port in the main body and the base. We listed all the required parts and the bill of material with the links where you can buy them on our GitHub page. You can also find the SCADs, STLs, and IPT files for the print. Keep in mind that the SCAD files uses the libraries from OpenFlexure and ThreadSCAD, so you need to download them and put them in one place. We also made very small simulation that shows Brownian motion of the trap particle and the forces acting on it. Now I will show you how to prepare a sample. For that you need a microscope slide, a cover glass, ideally cover slip number 1.5 because most of the objectives are optimized for that thickness, and a double-sided tape. We use the tape to create a small spacer on the slide. Then place the cover slip on the tape. This creates a very thin chamber between the slide and the cover slip. If you put a droplet of your sample at the edge of the cover slip, it will flow into your sample by capillary action. In this example, our sample is a small colloidal bead about 4 micrometer in diameter made of silicon oxide in normal tap water. Here is what the sample looks like under the microscope. The optical trap is on the left side of the image, and you can see the sample is not super clean because it's just the tap water. but it's good enough to show the performance of the trap. To drag an object with this optical trap, I'm moving the stage and the sample respect to a fixed trap. It's better to change the step size down such that with moving the stage, the particles are not escaped from the trap. By pulling the trap object through the fluid, you can get a feeling for the viscosity, for instance, how thick or thin the sample is. It's like having a tiny hand inside your sample. So far, optical tweezers was a research instrument. Most of applications of optical tweezers have been in the labs. A famous example is stretching a single DNA molecule and studying how proteins interact with it. Optical tweezers is actually good to measure forces about femtonewton, 10 to the power of minus 15 newton, up to hundreds of piconewton. That is it for this build. If you have any questions, drop them in the comments. We will do our best to answer all of them. Subscribe if you want to see more, and follow our GitHub page. We are working on a software that helps you how to calibrate it and measure forces applied to your home-built optical tweezers device. At the end, a huge thanks to the OpenFlexure team for this fantastic platform, and a special shout out to Moshtaba Nosratlu, Wfront Principle, and CometLab for their great valuable support.